Today we're looking at water and sugar transport in vascular plants. This is geared for Biology 11, SBI3U, and it's Mr. Roddy. Tonight, or this afternoon, we're talking about xylem phloem, companion cells, and how different materials are transported in plants. And if you remember a plant, if you look at a vascular plant, a simple sketch of it, I've got the leaf, the stem, and I'm just going to do some roots here. Okay, so we've got the roots down here, we have a stem, and then we have a leaf. Okay, so based on this, when you think about it, plants, their primary goal in the leaf is photosynthesis. Okay, and so when we review and think about photosynthesis, we need to think about looking at the calculation. The very general calculation is water plus carbon dioxide in the presence of sunlight. Okay, so in the presence of our sunlight, makes sugar or glucose and oxygen. Okay, so that's our whole premise behind when we start thinking about plants. Up in the leaf, you need water, you need carbon dioxide, and in most places in a terrestrial system, you have the water down here, and you have the CO2 up here, and you need that inside to do your photosynthesis inside. Okay, so you need to be able to import the carbon dioxide and grab the water and bring it up to the leaf. Okay, so we'll think about that. Let's just look at water for a minute. Okay, if we're thinking about water. Okay, water is polar, which means other polar objects will be attracted to it, and it will be attracted to other water molecules as well. And so, simply put, water sticks to itself. Okay, and to other water or other polar molecules. Okay, so if it sticks to itself, we call that cohesive. And when it sticks to other things, we state that that is adhesive. So those are some of the properties that you want to think about in terms of water. Being cohesive, it means it sticks to itself, and adhesive means it sticks to other things that are polar. Non-polar items do not stick, so you have to remember that we have to think about non-polar versus polar, but in this situation, the water's in the ground, and these properties will help it move up. Okay, when we look at the water transport, so we're looking at water transportation. And again, we're talking water movement within terrestrial plants that have a root system and a vascular tissue. And so the water movement is sort of based on three premises. The first is root pressure and absorption. Okay. Also, water, we talk about transpiration. Okay, so we have transpiration, which is up in the leaf, and then the properties of cohesion, tension also help play into the fact that water moves up the leaf. Okay, so let's start. Basically, we're taking it a very simple look at this. We're not looking at water potential or anything like that. All I'm looking at in terms of this is we look at how water moves from a high concentration of water to a low concentration of water. Okay, so we're going from waters moving from high concentration water to low concentration water. So if we had two cells or two entities, and one was fresh water and one was seawater or salt water, that would mean that the seawater has a higher concentration of salt than fresh water. And if you look at it there, that means this water has a higher percentage of water or concentration of water, and the seawater has a lower concentration. And so what would happen naturally through cell membranes, because water can transfer across cell membranes, is the water is going to migrate over to there. Water does that naturally. That's the osmosis side of things, okay? And so when you think of it in terms of a root system, I have my roots, pardon my inability to draw. Think of your roots sticking in the ground. And so you're underground here. If in the root you have enough solutes, okay, things are in the roots and the tissues, okay, and you have water outside and less solutes than inside, the water is naturally going to flood into the cells of the roots, okay? And as the water floods into these cells, they start to fill up, and they're going to transfer and move on to other cells. And ultimately, that water ends up getting pushed into the xylem. And then it will slowly work its way up, and there's what's called root pressure. And you have root pressure pushing on the water. So basically all you need to think about is if there's enough solutes down here and stuff dissolved in the tissue and there's water around it, not too much water, you don't want to drown it, but the water will flow into the root naturally. Not a lot of energy needed to pull the water in because water will cross over. As long as the root hairs are not covered in a waxy cuticle or things like that, as long as there's access for water to cross the cell membranes, they will cross and they will move into the roots, and the root pressure will start to push the water uphill. Okay, it's starting to even go against gravity right there. Okay, so in the roots, basically water is just flowing according to its gradient. From there, let's go up to the top of the leaf. Okay, and so up in the leaf, I'm just going to draw a die-cut leaf quickly. Okay, so if you did a cross section, you have your cuticle at the top, okay? You have your epidermis right underneath that, and just below that you have your palisade mesophyll layer, which is your site of most of your photosynthesis. And then you will have your vascular bundles here. Okay, we'll just do another vascular bundle. And then we have your spongy mesophyll. And then your lower epidermis with your guard cells and your stomata. Okay? And up here you have your photosynthesis taking place. Okay? So you need access to water. In the vascular bundles, you'll find the xylem. Okay? And at the end of the xylem, if you think about it, at the top here, if you have water at the top, the water is going to evaporate out into the mesophilic tissue, into the spongy mesophilic tissue. Okay, well, actually not the tissue, but into the air pockets around. Okay, so now you're getting water evaporating into this tissue. And remember, water goes from an area that's high water, which would be the xylem, into the air, which is lower water. And then ultimately, outside is usually less humid, and so the water is going to exit out through the stomata. So you now have water exiting out. And that is transpiration. Okay, and so transpiration is basically the water escaping the leaves through the stomata as it's evaporating out of the vascular tissue. And depending on which paper you're looking at or what research you're looking, between 95 and 99% of the water that is going up the root system that has been absorbed will end up leaving the plant as evaporation from the transpiration. Okay, so when people think plants are very efficient, well, they're not very efficient at moving the water. Well, they're efficient at moving the water, but they're not very efficient at keeping the water. Most of the water actually escapes out. The leftover bits, that's what feeds the rest of the plant so that photosynthesis can take place, that the vacuoles can stay full, and the cells can stay turgid. Okay, so that's transpiration. Now the point is when you get to the sort of in the middle, when you think, wait a minute, we've got the root down here, and we've got the leaf up here. How is it that the water will naturally go up the xylem? Okay, you got to think about xylem as being very, very thin, a thin tube, an extremely thin tube. And if you were to grab a drink, okay, let's make it a nice yellow drink, okay, and then you put a straw in there. If you just looked at the straw, the level of the liquid would actually be higher in the straw. If I put another straw in there that was even thinner than the original straw, well, the level of the water would be higher up. And that's just a natural reason being is that water is sticky to itself and it sticks to other things. So as it'll move up the wall, sticking to the wall, provided it's polar, and it'll stick to itself, so it pulls it up higher. Okay, and that's capillary action. And so the narrower the tube, the further up it'll naturally go without any force, anything else needed, it will naturally do that. And so when you think about when you're drinking out of a straw, if you put the straw there, the way you will get the liquid to move up is if you create a negative pressure at the top. And that's usually when we take a sip. But in a leaf, if you fill the tube with water, as high as it'll go, okay, and if you start removing water at the top, you're creating that negative pressure at the top. And because there's negative pressure, more water wants to go up. And because it wants to go up, it's going from here. So there's a tension pulling it apart. Okay, you're trying to pull it apart. But because water is cohesive, when this water goes away, this water will come and fill its place. And when this water gets pulled away, this water will fill its place. And it'll slowly move its way up until it gets to the end, and then it's evaporated out. And so when you look at cohesion tension, that's right in the xylem tissue, okay? Transpiration is the end product as it's leaving, and root pressure is where it gets started, where the absorption of the water happens, and you start filling the xylem tissue with water at the bottom, okay? So that's basically the movement of water through the plant, okay? So hopefully that makes sense. It's a pretty general basic look at it. You can ask questions if you want. Please feel free. We're going to move on to transpiration. Okay. And so we're moving to, or not transpiration, we're going to try translocation. And that's movement of sugars in plants. Okay. So an example, that might be moving the sugars from the leaves down to the roots. Okay, maybe they're storing it for later. Or maybe it's going down to a tuber to be stored. So like in a potato, think about a potato. The green plant is growing above the surface. It's taking that sugar and it's taking it down and storing it into one of the tubers as a potato, and it gets converted to starch. Okay? And when you're looking at this, again, I'm just looking at it very simplistically. You have your leaf that's producing its sugars. Okay, I'm just going to call it S. Okay. When you get into the tissue of the phloem tissue, okay, this is phloem tissue we're looking at. Next to the phloem, we have these things called companion cells. I'm just taking it as simple as possible. So we've got the companion cells, and then right beside the companion cells, we have the phloem tissue. Okay, and so the phloem tissue, again, is similar to xylem in many respects in that it's a tube in a, again, really general sense. So what happens with the sugars, okay, I'm going to make these sugars as S. We'll just do this. Okay, the sugars get pumped out into the companion cells where the concentration starts to build, and then it moves into the phloem. Okay, so now you've got the sugars there. Next door to the phloem, you have the xylem tissue. Okay, and so in the xylem tissue, you've got your water molecules being pulled up. Okay, so the water is coming up the xylem. Well, and we learned earlier, what does water do if it goes to an area that has less water? It naturally moves. So what happens is the water, some of the water actually goes into the phloem tissue. It dilutes the sugars, which also causes more sugars to come in, and that pressure that is created from the water going in, the phloem is now a conduit to take this sugary sap and send it down towards the roots. Okay? And if we're down in the roots for a second and we think about it, the sugar gets down here. Well, it gets pulled out of the phloem into the tissues to be stored. Okay, so we've got it stored in the tissues. Well, that means now we've got more sugar in the tissues, which is also going to cause the water to want to come into the roots. And as the water comes into the roots, it gets forced into the xylem, and the xylem goes up. The water that came down with the sugars will also end up being recirculated and going back up. So that's really a basic look at translocation as well. Okay, so we quickly went through water movement and we've looked at sugar movement. One of the next lessons we can look at are the stomata in terms of gas exchange as well, and when they open and close. All right, again, if you have questions, please feel free to email me, and hope this lesson was helpful.